Why is serology a cornerstone in medical laboratory technology, you may wonder? Serology is an intriguing field that dives deep into the essence of our body's defense mechanism. It's the scientific study of serum, the clear yellowish part of blood that remains after clotting. But it's not just the serum. It's about the intricate dance of reactions that our immune system performs when it encounters pathogens or introduced substances. Picture it this way. Our bodies are like fortified castles. The immune system, our knights and archers, springs into action when invaders like bacteria or viruses threaten the peace. Serology is the scribe that records these battles, studying the strategies, the weapons used, and the aftermath. This understanding of the immune system's response is pivotal as it forms the basis for diagnosing a myriad of diseases and conditions. From viruses like HIV to autoimmune diseases like lupus, serology holds the key. Understanding serology provides the basis for diagnosing various diseases and conditions. So what principles guide serological testing? Well, let's dive in and explore these fascinating concepts together. At its core, serological testing is all about antigen-antibody reactions. Antigens, which are typically foreign substances such as bacteria, viruses, or toxins, trigger an immune response in our bodies. Antibodies, those diligent defenders produced by our immune system, latch onto these invaders, marking them for destruction. Now, when an antigen and an antibody meet, they form a complex, kind of like a lock and key. This reaction is the cornerstone of serology. By observing these reactions, we can identify the presence of certain diseases in the body. These antigen-antibody reactions can take several forms, each with its unique characteristics. First up, we have precipitation reactions. These occur when soluble antigens combine with their antibodies to form large, insoluble complexes that precipitate or settle out of the solution. Next, we have agglutination reactions. This is when particulate or cell-bound antigens aggregate to form visible clumps when mixed with their corresponding antibodies. This reaction is particularly useful for blood typing. Then there's neutralization. This reaction happens when an antibody neutralizes the biological effect of an antigen. For instance, the neutralization of a virus by an antibody prevents the virus from entering a host cell. Last but not least, we have complement fixation. Here, the antigen-antibody complex activates the complement system, a part of our immune system that enhances the ability of antibodies to clear pathogens from the organism. Each of these reactions serves a crucial role in diagnosing a variety of diseases. They allow us to detect the presence of specific antigens or antibodies, thereby indicating whether a person has been exposed to a particular pathogen. So there we have it. From antigen-antibody reactions to the various types that can occur, these are the principles that underpin serological testing. These principles lay the groundwork for conducting and interpreting serological tests. What are some of the most common serological tests used in hospitals? Now that's a question that opens the door to a fascinating world of disease detection. Let's start with antibody tests, also known as immunoassays. These are designed to detect specific antibodies that our immune system produces in response to an infection or a vaccine. For instance, when someone is exposed to the HIV virus, their body produces HIV antibodies. An HIV antibody test can detect these antibodies and confirm the presence of the virus in the body. Similarly, antibody tests are used to diagnose diseases such as hepatitis B and C. Next in line are antigen tests. While antibody tests hunt for the body's response to an invader, antigen tests look directly for the invaders themselves. These tests identify specific proteins or substances associated with a particular pathogen. For example, antigen tests are commonly used to detect the presence of the influenza virus or the new coronavirus, SARS-CoV-2. Then we have the complement system tests. The complement system is a part of our immune system that enhances the ability of antibodies and phagocytic cells to clear microbes and damaged cells from our body. It also promotes inflammation and attacks the pathogen's cell membrane. When disorders of the immune system are suspected, such as lupus or rheumatoid arthritis, complement system tests can help confirm the diagnosis. Last but not least, let's not forget about autoimmune serology tests. These tests are designed to detect autoantibodies, which are antibodies that mistakenly target and react with a person's own tissues or organs. autoimmune serology tests are key to diagnosing conditions like rheumatoid arthritis, lupus, and type 1 diabetes. In a nutshell, these are some of the most common serological tests used in hospitals. From detecting viruses to identifying autoimmune disorders, these tests play a crucial role in disease diagnosis and are a regular part of hospital laboratory procedures. How are these serological tests performed in the laboratory? Now that's a question that unveils the fascinating world of laboratory science. The journey to a serological test result involves a series of precise and meticulous procedures. Let's start at the beginning with blood sample collection. This is typically done through venipuncture, where a needle is used to puncture a vein, usually in the arm, and draw blood into a tube. The sample must be handled with care to prevent hemolysis, or the breakdown of red blood cells, which could compromise the accuracy of the test. Once the blood sample is collected, it's time for sample preparation. This involves separating the serum, the liquid portion of the blood, from the cells. This is usually achieved by centrifugation, a process that uses centrifugal force to separate the blood components based on their densities. The serum, rich with antibodies, is then ready for the actual testing procedures. Depending on the specific serological test being performed, various laboratory techniques may be employed. For instance, in an ELISA test, an enzyme-linked immunosorbent assay, the serum is added to a plate coated with a specific antigen. If the corresponding antibodies are present in the serum, they will bind to the antigen, and a subsequent reaction with an enzyme-linked secondary antibody produces a color change, indicating a positive result. On the other hand, tests like the agglutination test rely on the clumping together or agglutination of cells or particles in the presence of specific antibodies. A visible clump indicates a positive result. From collection to testing, each step is a delicate dance between science and technique. Precision, accuracy, and strict adherence to protocols are the guiding principles. Remember, the quality of the test result is only as good as the quality of the process leading to it. Proper technique is key to obtaining accurate and reliable test results. So whether you're a seasoned laboratory professional or a budding medical laboratory technologist, always strive for excellence in your laboratory techniques. Test results are in. What's next? Interpreting serological test results isn't as simple as a pass or fail exam. It's a nuanced process that involves understanding the test's purpose, the patient's history, and the clinical context. Consider positive and negative results. A positive result doesn't instantly mean the presence of disease. It indicates that the patient's immune system has reacted to a specific antigen, which could be due to a current infection, past infection, or even vaccination. Similarly, a negative result doesn't necessarily mean the absence of disease. It might mean that the body hasn't produced enough antibodies yet, or that the immune response is directed towards a different antigen not tested for. Now let's talk about titers. Titers are a way of expressing how much of a specific antibody is present in the blood. The higher the titer, the more antibodies there are, indicating a stronger immune response. However, high titers don't always correlate with disease severity. Some diseases trigger a strong immune response but cause mild symptoms, while others trigger a weak response but cause severe symptoms. Context is key in interpretation. The patient's history and symptoms must be considered alongside the test results. For instance, a positive result for an infectious disease in a patient with relevant symptoms and exposure history is more likely to be a true positive. On the other hand, the same positive result in a patient with no relevant symptoms or exposure history could be a false positive. Keep in mind that serology tests aren't standalone diagnostic tools. They are part of a larger diagnostic puzzle. The results must be interpreted in conjunction with other laboratory data, clinical findings, and patient history. Remember, tests are only as good as the interpretation of their results. A well-performed test with a poorly interpreted result can lead to misdiagnosis and inappropriate treatment. So it's crucial to interpret these results accurately and holistically. Interpretation of test results is as crucial as the test itself in making accurate diagnoses. What challenges and considerations should we keep in mind in serology? Well, like any scientific field, serology isn't without its hurdles. For starters, false positives or negatives can occur. This can lead to misdiagnosis, causing undue stress or improper treatment. Cross-reactivity, where antibodies react with antigens not intended for the test, is another challenge. It can result in inaccurate results. Thus, confirmatory testing is often required, adding another layer of complexity. Let's not forget about considerations outside of the laboratory. Patient privacy is paramount. All test results must be handled with the utmost confidentiality. Quality control also plays a crucial role. Reliable results depend on well-maintained equipment, proper sample handling, and accurate record keeping. Despite these challenges, serology remains an integral part of medical diagnostics, playing a vital role in patient care. The road might be bumpy at times, but the destination, understanding and treating disease, is unquestionably worth it.